Researchers have developed the world's first 3D-printed brain tissue that grows and behaves similarly to natural brain tissue, marking a significant leap for research into neurological and neurodevelopmental disorders. This new 3D printing technique utilizes a horizontal layering approach and a softer biological ink, allowing neurons to interconnect and form networks resembling human brain structures. The ability to precisely control the types and arrangements of cells provides unprecedented opportunities to study brain functions and disorders in a controlled environment, offering new avenues for drug testing and understanding brain development and diseases such as Alzheimer's and Parkinson's. The 3D printed brain tissue can form networks and communicate through neurotransmitters, similar to interactions in the human brain. A leap forward in neurological disorder research. This new printing method allows precise control over the types and arrangements of cells, surpassing the capabilities of traditional brain organoids. The technique is accessible to many laboratories, requiring no special equipment or culture methods, and could have a significant impact on the study of various neurological conditions and treatments. This could be an extremely powerful model to help us understand how brain cells and parts of the brain communicate in humans, says Su-Chun Zhang, professor of neuroscience and neurology at the Weizmann Center at UW-Madison. Our tissue remains relatively thin, which makes it easy for neurons to readily receive enough oxygen and nutrients from the growth environment, says Yan. It could change the way we view stem cell biology, neuroscience, and the pathogenesis of many neurological and psychiatric disorders. The technique is accessible to many laboratories. Previous attempts to print brain tissue have had limited success, according to Zhang and Yuanwei Yan, a researcher in Zhang's lab. The group behind the new 3D printing process described their method in the journal Cell Stem Cell. Instead of using the traditional approach of 3D printing, which involves stacking layers vertically, researchers went horizontal. They placed brain cells, neurons grown from induced pluripotent stem cells, in a softer gel than that used in previous attempts. The tissue still has enough structure to hold together, but it's soft enough to allow neurons to grow into each other and start talking to each other, says Zhang. The results speak for themselves, meaning cells can communicate with each other. Printed cells reach through the medium to form connections within each printed layer as well as between layers, forming networks comparable to human brains, neuroscience news reports.